I should mention that Tijana is basically a virologist and mechanistic biochemistry. Well, that's a new term. You'll have to explain that to us. Unraveling the molecular mechanisms of large macromolecular translocation across biological membranes. Big things going through small holes, I guess that is. She did her PhD in virology at Harvard Medical School, working with Max Niebert, and she has worked mainly on membranes. But I think what's interesting is that in 2011, Tijana relocated her research from Harvard Medical School to the University of Colorado, and she's got here in parentheses a move dictated by family considerations, parentheses. So we'll have to hear about that too. But she's won a LOreal AAAS Fellowship for Women in Science, and she's also a board member of the society. So,
thank you very much for the introduction, and I very much appreciate the opportunity to speak at the meeting today. And I think the invitation couldn't be more timely in that I'm precisely at a point in my career where representation of women in my field in academia suddenly drops. So I'm a senior postdoc in biology, and women are not underrepresented if you look at PhD students or postdocs. But if you look at faculty appointments, their numbers or their proportion suddenly declines. And so why is that? So there's a lot of theories and a lot of opinions, but I'd like to address this point through my own experience, and precisely because I'm exactly that point in my career, and I'm trying to decide what the next step will be, and I'm not so certain of it. And so I would, and to put my current state of mind in context, I want to share with you a quote from my personal statement to Harvard University back in 2002, when I was applying to PhD programs, to PhD program in virology. And so what I wrote at the time was, although the road in front of me is just beginning to unfold, the path I traveled so far was undoubtedly one that gave me enough experience and knowledge, sufficiently tested my endurance and ability, and brought me to the point where I can state with certainty that I see my future in research. And if you go on to read my statement, I really talk about academic research. And 10 years later, or more than 10 years later, when I'm, after a ton more training, when I'm more in charge of my research than I've ever been, I could say I'm the master of the research that I do. And I love it more than ever. I'm not so sure what happens next. and so how is that? So I'll tell you a little bit about the science that I like to think about and that I work on. And then I'll put it into personal context as I try to address this point. So I studied viruses for almost 15 years now. You can't talk about viruses for a very long time without starting to think about cells, as viruses absolutely depend on cellular resources for their own survival and replication. But one of the first roadblocks they encounter on their way into the target cell is the lipid bilayer barrier that surrounds each living cell. And so they have to cross it, and they've evolved mechanisms to do that. And the virus I study in my postdoc is influenza, and it's a part of a large group of viruses which are themselves surrounded by lipid bilayer. And they cross membrane barrier by mechanisms which promote fusion between the viral membrane and the membrane of the target cell. And they accomplish this task by viral proteins which are expressed on the virus surface, decorate the virus surface, and serve as fusion machines. So the fusion machine of influenza is called hemagglutinin, or HA. And you can see it as this protruding spike on the virion surface, densely covering and occupying most of this surface. It's the most abundant protein on the virion surface. And so influenza on its way into a cell, engages the cellular receptor. It is internalized into these membrane-bound vesicles called endosomes. And here, the naturally low pH of this compartment serves as the trigger for membrane fusion. So influenza has evolved to use this low pH as a cue that it's at the right place at the right time, and it can unfold its fusion program. And so I don't mean to go into details of this transformation. I just want to point out that hemagglutinin is believed to undergo a series of structural rearrangements that are believed to be shared among all envelope viruses, those that are surrounded by lipid bilayer. And through a series of really large structural transformations, the fusion proteins make a physical link between the viral and target membranes and essentially bring them together. And so although we tend to draw these models in quite some detail, what is really known about this process, and hemagglutinin is the best studied viral fusion protein of all fusion proteins. What we really know about this protein is what it looks like before fusion and what it looks like after fusion is completed. And so even though we tend to draw this detailed molecular pictures of what happens in between, this is really largely an area of a gray zone. What happens in between is inferred from these two structures and from indirect biochemical evidence. And so with my postdoctoral work, I set out to add color to this gray area and to provide direct evidence for some of these structural transitions, and moreover to add a kinetic component to these structural transitions. And to do that, we needed a fresh approach. So I joined the lab of Steve Harrison, and we worked in collaboration with the single molecule biophysicist Antoine Vanoyen at Harvard. He was at Harvard at the time. He's now at the University of Groningen in Netherlands, but we continued the collaboration. And we decided to study this process of membrane fusion at the level of single individual virions. So rather than looking in a classical way at averages of entire populations, we wanted to look at individual virion at a time in order to uncover these kinetic details. To this end, we needed a sort of fancy microscopy technique called turf microscopy. It's essentially a way of illuminating the sample, a fluorescently labeled sample that allow us to visualize, to have high signal to noise ratio and to visualize individual virions in the process of fusion. And so here is what this experiment looks like under the microscope. So in the bright white dots are virions, and this is them fusing later. You see them because their membranes are labeled with a fluorescent dye. With trigger fusion by lowering the pH using a microfluidic flow cell system of the surrounding environment. And once the fusion starts to take place, viral membrane starts to mix with that of the target membrane, and the dye that was initially constrained in the virion is released into the target. And so you see that as exploding. This movie is just looping. Looks like exploding stars. As individual virions are fusing with the membrane. But now, because we can see them one at a time, we can also analyze them one at a time and derive these frequency distributions, which are basically statistics of entire populations, but representing each virion individually. And it is this kind of data that suddenly can teach us at a level of detail about this process that wasn't accessible before. And so this was all very exciting. I joined the postdoc. There was a whole new world that was opening up for me. I was trained as a classical virologist. I was starting to enter the world of biophysics. I was learning how to devise molecular models from statistical data. I was learning how to program, both to analyze this data and to devise computer simulations to test our kinetic models. So there was a whole new world that was that I was discovering. But if it were simply a matter of getting the work done, it wasn't that simple. So life got in the way. My husband got a faculty position, faculty offer at the University of Colorado Boulder. I was well into my postdoc, so that I wasn't ready to leave it, to drop what I was doing and start something new. But I was still years away from being done. And so we had very limited choices. Either he doesn't accept his position and he only got one choice, and shoots himself in the foot, and in terms of progress and his own career development, or we stay apart for a number of years. There was a third possibility that crossed our minds, but we didn't dare to suggest it to anybody. But my incredibly supportive advisor, Steve Harrison, decided to offer, and he suggested that I go and join my husband at University of Colorado, and he would continue supporting me. I would continue my work remotely, and my salary would keep coming from Harvard, and we would have Skype meetings to discuss our data. And so it sounded great, but there was a caveat, and that is there was no microscope at the University of Colorado that I could use that would meet my demands, especially when I used to do this experiment, I spent 12 hours in front of the microscope on a given day, and there wasn't an option for me at the University of Colorado. These microscopes, commercial microscopes, cost on the order of $250,000. So as a postdoc, that's sort of an impossible goal one can try to reach. And of course, my advisor couldn't do that either. No matter how hard he tried to support me and wanted to help out. So, okay, so the plan was, we'll figure out a way, we'll make it happen. But let me try to get a bit more work done so I get in a better position when I'm looking for money to buy a microscope. My husband delayed start of his appointment by a semester. I delayed joining him by another year. My work was starting to look exciting and great. But I was pregnant, and when I was about to be, just before I was 9 months pregnant, and there would be complication to, I would need special permission to fly, I decided to join my husband. and I moved to Colorado in November. My baby was due December 24, next month. I moved, here's me with a bunch of boxes behind me. And I learned about this wonderful opportunity, the LOreal Fellowship for Women in Science. Deadline was December 12th. I was just hoping my baby would not be early, and she listened. I submitted the application. My baby was late. Maya was born on January 4. She was almost 2 weeks late. So my application was in. I had some downtime during my maternity leave. I decided to get a couple of papers out of the way. So I submitted in March 2011 sort of a leftover work from my PhD times with my advisor Max Nybert and collaborator Steve Boulan. We finished revising it, submitted it in March. It was published the same year in the journal Traffic. I wrote one of the first drafts of my side postdoctoral project that I managed to complete before leaving Harvard. It took some time to submit this and get it published. I think, yeah, it was published in 2012 eventually. The last day of my maternity leave, I attended a conference, the National RC Conference in Denver. I like to show this picture because I make a joke in them. Oop, what happened? So it just died. So just to give you a background, the photo I'm about to show. So it's a national RC conference. I came as a representative of the New England branch from Harvard. I belonged, and my funding support from Steve came from the New England RC branch, or any RCE, or nurse. And so I like to call this a photo nursing. So my maternity leave was over. I went back to work. Soon after that, I learned that I was awarded one of the 5 LOreal Fellowships that year, 2011. And I couldn't be more grateful. It literally made possible what seemed kind of desperate at the moment when I moved. The award is, and I want to say not only did I get the award, but they were so amazing that they allowed me to get the entire award sum up front rather than getting it into installments. They also allowed pre-award spending, so I could immediately start buying microscope parts and not wait for the award to start, so I didn't have that delay. So quite amazing experience and support. The award was a little short in terms of the sum of money in making it possible to buy a commercial version of the microscope. And so I basically needed to build microscope from components because that's much cheaper. So I went from a user of microscopes, you know, us biologists, this is what we typically train to do, push a button and hope it works. When it doesn't, you call for help. So I had to learn how to design and build one and to do it from individual components, and then to heavily negotiate each single part that I was buying because I had more time than money at a time. And I managed to get the entire microscope to cost under $80,000. So, okay, I did it. I was very happy about that. And now it's finally simply a matter of getting the work done, or not so simply. So here's me trying to work from home a little bit with my daughter Maya on my side. And here's Maya getting her first experience at push biking in the long corridors of my research building after hours. You must admit it's pretty impressive corridor. While I'm in the lab trying to squeeze in, finish the last experiment of the day. And so all this work paid out. We started to add color to the gray zone of the model I showed you at the beginning. We provided direct evidence for existence of a number of intermediates of fusion that I showed you. We identify a very long-lived intermediate, that we think is a very attractive target for potential inhibitors. And we came up with a model that, with an integrated molecular movie of events that happen in the virus target membrane interface. So I don't want to go into details, but basically we described a model and a way in which action of multiple hemagglutinin molecules is coordinated to mediate a single fusion event. And finally, this work was published in one of the first issues of the new journal eLife, and it was profiled in an official Harvard newspaper, Harvard Gazette. And so I got to the point. So I did what I came to do, what I wasn't sure was possible to make happen. It happened, and so why am I not feeling more empowered than ever to keep going as I should? And so this has to do with my experience at CU, and it was Eileen's article in New York Times that I read, where I first ran into this quote. But this quote was originally from an article in Washington Post by Meg Urry, and where she described a modern day discrimination in the United States. It's no longer in your face. Nobody's going to tell you can't do it because you're a woman. But there is a slow drumbeat of being underappreciated, feeling uncomfortable, and encountering roadblocks along the path to success. And so, just with a few examples, I just want to describe my experience at CU. So before, oops, it died. So before being before even moving to CU, before having an opportunity to summarize in one sentence what I do, let alone show somebody my CV, or let alone having had the opportunity to to show what I can do. I was told by a faculty who was to become my formal host at this institution that the best I could hope for is a research faculty appointment at CU. And I said, you know, I don't have to limit my choices to CU, and my aspirations are not to become research faculty. I'm aiming for a tenure track faculty. Well, you know, the kinds of candidates we see. And so it really outraged me, because he doesn't know, how does he know my limit, my cap, without knowing anything about what I do? So that there had to be some underlying assumptions. And there had to do something with the fact that my husband was a superstar with a faculty appointment, and with the fact that I'm following. So either, you know, the assumption is, you know, if you have one superstar, what's the probability of both spouses being superstar? Not very high, maybe. Why not? Or something to do with the fact that, you know, his career is clearly dominant if I'm, you know, the one following. It's just the way timing worked out. It's not how either one of us, my spouse and I, feel. We both acknowledge that at different points in our careers, either one of us could be making sacrifices. And so this I heard repeatedly over and over the next 3 years, even after I had a chance to show what I can do. I continuously got meeting expectations, so the average evaluations by my local host. And initially I didn't say anything, but this year, you know, third time in a row, I decided to confront him and ask him why, to you know, rationalize this, to justify this for me. And he said, you know, he couldn't be bothered because, you know, I'm not really working with him, and so he can't really evaluate me. But I pointed out that I'm physically in his lab. I'm attending lab meetings and presenting in them. You know what I publish, you know, the words I got. So what's happening you from being in a position to evaluate me? And he said, no, it's just a formality that I'm signing this, and don't worry about it. And I said, but it's a public record, and you know, I don't like to have meeting expectations, and I don't agree with this. And he said, okay, well, write a rebuttal letter, explain why you don't agree. And I said, why don't you just call my advisor at Harvard and ask what he would evaluate me? No time for that. This form has to be turned in. So encountering roadblocks along the path to success. So I was never, so thankfully LOreal Fellowship existed, and I could apply for it as an individual from a research institution, but I was never able to apply for any of the standard postdoctoral NIH grants. I started writing K-99 proposal, and midway I learned from Harvard's grants office that they would not submit my grant citing liability concerns because of my remote location. At CU, I first didn't have status to be able to apply, and when that was worked out, I realized that I really have no credible mentor there to justify the mentor phase of the project, of the grant, and so I never was able to apply. Feeling uncomfortable. So I decided last year to apply for faculty positions, but my e-life paper was running late for regular searches. Although it wasn't late for CU because I could update them. I was there. I knew who the search committee members were, and I could tell them, hey, this was published. I applied to regular search, but they decided to single out my application and make me go through a spousal hire track. And this just means that the engineering dean, the School of Engineering is where my spouse belongs, where my spouse is faculty, would create a position, faculty line for me if I'm good enough to get it. And I was assured that this is the same kind of treatment as any regular interview process. Don't worry about it. You're not going to be considered any of a lesser candidate because you're going through this path. And I was finally convinced to do this. When I was invited for an interview, department chair basically told me, I'm organizing your interview. We usually take our candidates out for dinner. I was not planning this in your case, but if you want to go to dinner Friday night, let me know. So invite yourself if you really want to go. Well, I wasn't that desperate. I didn't know. You know, I was kind of stunned. I didn't know what to say. I basically said something about, you know, having additional interaction with different faculty would be beneficial, but, you know, I'm not going to push for it, and it didn't happen. I was misled by my interview host. Ahead of the interview, he told me to for my chalk talk, or the proposal part of my talk, he told me to prepare to discuss one or more specific aims for a potential NIH or NSF grant. When I got there 2 minutes before my chalk talk, he was told, okay, so part of your talk is going to be about research, and the second part is going to be about teaching, right? And I said, that's not what I was told, but okay, I'll improvise something. During my interviews, one of the senior faculty members basically asked me to tell me a little bit about myself because they actually never circulated my CV. He knew this had to do something with my spouse having a position in engineering, but he wasn't quite sure. And then finally, after the interview, I decided to interview a bunch of faculty members, ask them, you know, to give me honest feedback on the interview process. And one of the well-meaning faculty members basically referred to my interview as a pseudo-interview. Which kind of sealed my suspicions from the experience that I had. I didn't get an offer, and I'm still there. I didn't apply for faculty appointments this year. I was a little bit discouraged by the previous experience, and I wasn't sure if I had enough on my CV to get one. So I decided to take a year to try and push one more paper out and try again the next year. And if I don't succeed, try something else because I have to get out of CU and that environment. And so with this, I'd like to thank you for the opportunity to share my story.
I think the problem that we all face is that your story is not unique. You are part of a generation that is facing difficulties in getting employment, and it's quite amazing because I think you're probably roughly in the cohort of my daughters. And it's interesting how they are living their cohort and themselves, living contract to contract, even though they're producing extremely good work, just as you are doing. And I think this is a dilemma that we will have to address in our discussions because it's very clearly destructive for our country. It's destructive because it means that we're losing a whole generation of brilliant researchers, men and women. And we're living in, as was discussed earlier, the funding situation is going to, I think, cause the United States to go from being the creativity leader to be the creativity follower. And ironically, what we're doing is our institutions, no one's talked about it yet, maybe Jan might be able to speak to it or others. The institutions are increasingly bringing students from overseas for the tuition, which is out-of-state tuition, which brings in the revenue that they're not getting from the state or getting from the federal government. And those students can't get visas after they finish, and they're going back to compete against the country. So we are training our own competitors instead of bringing them to our country. So you represent a challenge for us to take on to assist you and your cohorts, because otherwise we face a very bleak future. Questions? Yes.
I guess I have two comments or two thoughts. One, do you have fellow postdoctoral friends to interact with? I think in this very challenging time that you are, I think this transition is one of the most challenging times. It is very useful to have people to say, give you reality checks to support you, not necessarily among the faculty. So for me, that was really important. I also want to share one anecdote since you're at CU. I was an undergraduate there, and around that time, they hired Carol Check in biophysics, and she had a spousal hire, which was Tom Check, who then ended up going on to win the Nobel Prize. But
Right.
Tom Check was a spousal hire.
Yeah, I was aware of that. That's, yeah, somebody told me. It was one of the arguments they used to tell me, don't worry, go for it, it's okay. In terms of having fellow postdoctoral support, I think that's a great point. And the point is, I have a lot of friends, fellow postdoc students back at Harvard. I'm quite lonely at CU, because and it probably has to do with the way, you know, my unusual situations created the environment where, you know, I'm working in the corner on my own thing. I'm not really a part of anything, and I don't belong to anything bigger. So I'm more friends with my husband's faculty colleagues than my own peers, and that is a big problem.
To sort of follow up on that, I know, and I can't be the only person who's felt this. Sometimes when I go to meetings, I feel very lonely too. Or, I mean, when you go to a new situation, you don't have someone to talk to. And I have found it's made a huge difference at times in my career when I find the other lonely person in the room and end up talking to them. And so I guess your situation in Colorado is you now have a child, and which makes it more challenging, but I'm sure there are other fellows that actually are lonely too, and I have found several times in the context of going to a conference where I reach out to the person who's standing alone by their poster or not participating, that I've benefited a lot and that they were in the same boat.
Thanks. Yeah.
So I had occasion once to visit a university in Colorado, and the department chair asked me what he should be doing to recruit women faculty. And I sort of threw the question back at him and asked him what he was already doing. And he basically said, well, we're not doing anything. And we had a nice chat and everything, but I left unconvinced that things were really going to change there. And then I ran into this young woman about half a year later, and she told me she interviewed at that university. And so I asked her how it went, and she said, well, it went pretty well. She said, but you know, my husband and I both want jobs at the same place. And I said, so what happened after that? Did they give you a shorter timeline to make your decision? She goes, how did you know? So anyway, she ended up at a much better university with her husband. And now several more years have passed, and they finally hired a woman. So I'm wishing her all the best. But, you know, it just takes some universities a while to sort of get around to really understanding what they need to do to change. And I think, you know, you're unfortunately just caught in the wrong spot at the wrong time.
Thanks.
Yeah, so I'll echo that comment a little bit. So I'm from Cornell, and so circumstantially, Ithaca and Boulder are kind of somewhat similar. They're kind of isolated. They're kind of the only game in town, and Cornell struggles a lot with these kind of spousal hire two-body problems. But to their credit, I think they've been very proactive. But that's a comment, not my question. My question has to do with the postdoctoral mentorship at Harvard and whether the postdoc office there was in any way helpful, particularly with regards to the situation that arose when your husband got the offer. In our experience, one has the most leverage you ever are going to have when you have one of the spouses with an offer but hasn't said yes, and that is the time to negotiate everything. And so we've seen it play out a number of ways where it just happened recently, where we made an offer, we asked over and over in every legal way possible whether there was anything else we could do, and the person accepted, and then as soon as they got to campus said, oh, by the way, my spouse also is interested in an academic position. And we all just cringed because
Totally.
if they had told us that before, if that person had told us that before they accepted, we could have done a whole lot, but afterwards it's really hard. So did you get any advice along those lines from Harvard while you were there?
So the advice I was getting was mostly from professors, not the postdoc office. I didn't know to ask them for advice. We definitely made a few mistakes right off the bat. We did ask for what they can do for me, and the extent of what I got promised was a letter by the department chair, which said that they would provide work environment for me, meaning I will occupy somebody's lab. But we never asked for any funding for the microscope. We asked for funding for the microscope after my husband accepted and after I was about to move, which was too late, and they said no. So yeah, we definitely made mistakes, and that was the time to negotiate these things, and we didn't. We could have created a much better situation for me up front, and we just didn't know what to ask for. Or what we can ask for.
Well, I think that's the key. I think you can ask for anything, right? And if there's a message that I'm getting, everything's negotiable. And from what I've absorbed over the day, one of the things that I think we need to collectively do is impart that message on women early in their careers, that to not be afraid to ask for things. Not only to not be afraid, but their male counterparts are asking for those things. And if you are not, you are behind.
Yeah, I learned from my own mistake.
What is your department?
At Harvard?
No, at UC.
MCDB.
Because the chair of cell and developmental biology, Wendy Macklin, is someone you should probably turn to.
Okay.
I met her first when she was a junior faculty member. No, she was a postdoc at the Shriver Center in Waltham when I first met her, and I was a postdoc at the same time. So she's been there for only a few years.
Okay. So the current chair of MCDB, which is Molecular Cell Developmental Biology, is Mark Winey. But I'll look up the
Wendy
person.
Macklin.
Yeah, Wendy Macklin. Thanks.
I just wanted to say, just following up on the comment, now that you've missed the sort of pre-acceptance opportunity window, I think something that definitely works is that if your husband is doing really well, he can threaten to leave. And he definitely got to try it. I have seen a lot of very successful attempts like that a faculty member in a really good situation threatens to leave, and either the university comes up with a position for the spouse, or they just both move to a different university, and that has been a very good story. They have succeeded enormously. So I think really it's time for him to make the sacrifice for you.
They don't want to do another search. They're not going to want to do another search to replace your husband.
Yeah, so our plan is to apply next fall, both of us everywhere. So this last time was only me applying, and it was a little bit early. But next time we're both applying everywhere, and we'll go from there. Yeah. We have to have an offer first, yeah.
I think you're getting plenty of good advice.
Yeah, thank you very much for all the feedback.
And just a final comment, Colorado's not done very well with its football team either.
Oh.
It had suffered a football scandal, so. Any final comments? Because I think what we'd like to do is show those 2 very short videos, and then we'll break for dinner. Do you want to set them up? Thank you very much. That was great.
Welcome to Science Class. I'm Tom McFadden. You're about to see the story of Rosalind Franklin versus Watson and Crick as told by some incredibly talented 7th graders from Oakland, California. If you have any questions about the science, the content, the history, write them in the comments below. And if you stick around till the credits, I'll be back to give you some more information about how you can get involved. Enjoy.
Tom, will you tell us the science story?
Here we go.
Okay, I'm Jenny Watson, and I'm Francis Crick. Crick. And ain't nobody fresher than Watson and Crick. Crick, 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 crick. Nobody's making models like Watson and Crick. Crick, 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 crick. Cephalitis, but we beat them to that double heat. That double heat, that double heat. DNA. Oh God, don't tell nobody, but we're gonna solve DNA. Hereditary molecules under our name. We're making models, man, doing things our way. But these phosphates, we had them in the wrong place. Then Maurice gave us that new data. Newer model had to change. Shargoff and his roots say CGTA, double helix, true man, you know 2 chains. Skirt. Saw Rosie losing. Scientists are thrilling, man, that feel it when you go and start revealing life secrets if you're willing. But don't say that we were still when Rosie's dead, cause she never have seen. Now we're rich, famous, with no more prizes. Man, it's good to be Watson and Crick. Crick. Uh-oh, Crick comes, run!
Too many X-rays that I diffract, so it's not just gonna happen like that. Let's recognize Rosalind Franklin, all eyes on Rosalind Franklin. You show my data behind my back, so it's not just gonna happen like that. Let's recognize Rosalind Franklin, all eyes on Rosalind Franklin. Ooh, that's my pick, that's my pick. Ooh, that's my pick, that's my pick. I heard that you were showing my pick and you didn't think that I would find out. Then you wrote this nice little book and everyone fired up. Well, I'm back from the dead and I read what you said and I'm here to set the record straight now. Now when you learn about the W, you better also learn about me.
Dang, Maurice also did it? Whatever, I'll do it anyway.
Yeah, I'm talking her patient, yeah, I'm talking Curie. I'm talking Shirley, yeah, ladies, I'm talking me. Wanted to be a scientist since age 15. Became a world-class one, yeah, I follow my dreams. Maurice and I kings, we're going for DNA. She needs to change her hair, was all that you could say. While you were chasing models, I used my X-ray. But what you know about getting your data the hard way? And I showed it was a helix with phosphates on the outside. Calculated helical dimensions, and without my data to play with, you would have taken ages. I was almost there, you could read it in my pages. It has not escaped notice that you're a jerk. Should've got a Nobel for my work, but then the eyes will never know. Not till it's recognized, but not a female scientist, I'm a scientist. Man, look at my work and say, dang, that's a nice pick. Let me hear you recognize Rosalind Franklin, F-R-A-N-K-L-I-N. Recognize Rosalind Franklin, F-R-A-N-K-L-I-N. Recognize Rosalind Franklin, F-R-A-N-K-L-I-N. Recognize Rosalind Franklin, F-R-A-N-K-L-I-N.
Thank you for watching. I don't need to tell you this, but if you subscribe, you'll be able to see the future installments of these science history rap videos. And more importantly, if you click on the link in the description below, you'll be able to go to my blog where I have instructions about ways to kind of get involved at the next level. You can get the source material that we use to create this video. You can annotate the lyrics on Rap Genius. Or eventually, I'll be posting information about how to remix this material. If you do something cool, let me know about it on the blog. Thanks again. See you next time.
Very good. That's giving us a different look on what we need to do. The next video we're going to show you is something that has been put together with the staff at Genetic Engineering News. Our oldest publication? 32 years of public. Marianne Lee Britt Incorporated has published Genetic Engineering News. We obviously work very closely with the staff in the same office and worked with them in getting some wonderful bios of women in science for this piece. And we thought we would share it with you, which is also on the Gen website. A board women